Today's workshop is an ESTCP tech transfer effort. It is a FAQ, or frequently asked questions, on perfluoroalkyl and polyfluoroalkyl substances, otherwise known as PFAS. I'd like to introduce my co-principal investigator, Jennifer Field, who's the leader of the pack from Oregon State University, Jason Conder from Geosyntec, and Chris Higgins from Colorado School of Mines. So the way we're going to do this today is we're going to take turns answering the 10 FAQs that we've identified. But first, I'd like to refer you to the project number. It's ER 2015-74-T2. And if you visit the SERDP ESTCP webpage, you can download important deliverables from this project. So with that, these are the 8 questions that we're going to answer for you today, and Jennifer is going to lead us off with the first question. Jennifer?
It's my pleasure to leave off on the first question, and that is, what are per- and polyfluoroalkyl substances? So first, take a look at the cartoon in the upper right-hand corner. This is a molecule many people know now. It's PFAS, or PFOS, perfluorooctane sulfonate. And that cartoon is showing you some really unique attributes to many of the chemicals in this class that we're calling PFASs, or per- and polyfluoroalkyl substances. PFAS is a particular example, and it's one of those for which we have an EPA health advisory level. You'll hear a little bit more about that later. If you examine this molecule, you'll see that you very quickly pick up the carbon backbone that is entirely set with fluorines. There's no hydrogens on that carbon backbone. You also see the other attribute, which is the sulfonic acid head group, and we've drawn that in the water. And so here we have this fluorinated tail, a highly fluorinated tail sticking up out in water, and this polar head group in water. And this is showing a type of behavior for which PFAS is well known. You can see that that assembly of carbon and fluorine bonds has given it rise to a pretty unique nature of a molecule. So that carbon-fluorine bond is super strong. It's the strongest in nature. And when you assemble a number of those together, you get interesting properties, both from a functional point of view. So for example, the oil and water repellency that you enjoy in articles in commerce, textiles and papers and such is in part due to that, large part due to that fluorinated tail. On the other hand, some of the properties become a little less predictable in the environment and in the laboratory environment when you have so many carbon-fluorine bonds assembled together. And PFAS is an example of a perfluorinated compound. It's a pretty strict definition. It means that all those carbons have only fluorines and not hydrogens. And so this is why we're advocating a change or an awareness in terminology. If you look at the perfluorinated term, it really means something quite specific. But if you go to the molecule in the lower part of the page, this is another chemical that's found in mixed situations where we have an aqueous film forming foams, which is the subject of today's webinar. And you see that this fluorotelomere sulfonate has a different attribute. There's 2 carbons that have blue hydrogens on them. What you're seeing is a polyfluorinated molecule. It's not strictly perfluorinated. Now, is this just semantics? No, it's not. It turns out that those 2 carbons that have hydrogens on it represent a weakness in that molecule. And it turns out the fluorotelomere sulfonate is biodegradable, and you can also oxidize it. And that's very much in contrast to PFAS, which cannot be easily oxidized and is not biodegradable. So that small distinction in the chemistry actually has implications when it comes to the environment. And so right now, what you see is an international acceptance of the use of per- and polyfluoroalkyl substances for any settings when we actually have a mixture of things like PFAS and PFOA, which is perfluorooctanoic acid, and things like the telomere sulfonate. When they're all there together, we really can't use that term up in the upper right corner, perfluorinated chemicals, because it really doesn't work. So if we do take a closer look at PFAS and Pfoa, what you'll notice, of course, is they're anionic. They're anionic at all environmental pHs and physiological pHs. That means you and me. And what I mean by that is it's a pH range of 4 to 10. These are very strong anions. So what does that mean with regard to their behavior? Well, first of all, those strong acid groups are deprotonated. They're an anion. So that means that the molecules to which they're attached are not volatile. This is why we can't use things like gas chromatography for their measurement. This is why we can't airstrip them out of water. So they're really anionic. And also going right hand in hand is they have very high water solubility. That's why we see them transport readily in the environment. And that's why we see them pass through sediment in soil and into groundwater. Now, quite a few people might be wondering, why are they emerging just now? We've used aqueous film-forming foams for quite some time, decades, going back to the 60s. Well, the problem is just what I told you. They are anionic, so we couldn't see them with gas chromatography mass spectrometry, which was the common tool used to characterize sites and look for volatile priority pollutants, like you might be familiar with PAHs and PCBs and dioxins. So when we used this tool for early site investigations, we couldn't see them. However, there were quite a few field reports. People would say, wow, you know, the groundwater foams when I'm collecting it, or when it rains here, the soil actually foams. These were all important clues that we had persistent chemicals still present in these systems, but we overlooked the significance of foaming groundwater and soil for years. And it wasn't until the advent of liquid chromatography, tandem mass spectrometry, also known as LC-MS, it wasn't until we got stable benchtop scale models that more and more labs could use that we could see these materials. And that instrumentation has only been around for about the last 15 years. And then now everybody is increasingly aware you need high quality standards to do this kind of analytical work. And the standards necessary to do this work have really only become available within the last 10 years. So we want to move to question 2, and that is, what are aqueous film forming foams? Well, it's a specialty product. It's a very specialized, concentrated commercial formulation designed to do something very, very well, and that's put out hydrocarbon-based fuel fires. So if you have a tanker rollover, if you have an air crash, these are the materials that are coming out. What's important is to appreciate the broad, wide, wonderful world of fluorinated chemical production. And of that large sphere, AFFFs only make up 3% of that market. And so we're talking about a small slice of the overfluorinated chemical production, and that comes up later when we start talking about other sources. If we look in that 3% slice, you can actually see that 75% of that is used by the US military. And another 25% is used by very important markets, the oil refineries, municipal airports, municipal fire stations, and hydrocarbon fuel tank farms, anywhere where they store, besides oil refineries, lots of fuels. And so these often are fence line partners. You might see a military site and you'll see co-located, for example, municipal airports or oil refineries. So when we think about AFFFs, it's a complex mixture. It is proprietary, and so it actually took SERDP funding, the funder of this webinar and of this research, to allow us to look inside and understand what the composition of AFFFs actually are. In addition to highly fluorinated substances, the PFASs, there's also hydrocarbon surfactants and solvents like butylcarbitol. So it's a complex mixture and quite concentrated. Those PFASs are in percent levels. We don't often think about percent in the environment. That actually means in the container itself, it's grams per liter. So that's a pretty hot mix, and that's why you don't want to accidentally spill this around. So what's the history of these materials? Well, the US Navy commissioned the production and the creation of aqueous film-forming foams to put out fires back in the mid-60s. And so from the mid-60s till the early 70s, 3M was the sole source supplier of AFFF. And you can find the US mil-spec history for AFFF on the web now. It's kind of interesting reading. And what you'll see is that by the early 1970s, National Foam and Ansell, and even in the 1990s, we have other companies qualifying these AFFFs according to mil-spec and placing them on the qualified products list. And that products list is still available today, and it's very active, and we have the ability to purchase those materials off of the QPL. Please note that those were all requalified, I think, by the end of 2016, and so we have now even more players on the QPL. What this means, if you're a site owner, is that there's a very high likelihood, and we often see quite a bit of evidence when we go to any particular site, the calling card or the characteristic features of one or more AFFFs within a single site, and that's because of this history. So how was AFFFs used on US military sites? Well, it turns out there's a number of activities, all of which were prescribed. And that was, for example, in the top row, capacity testing. This was the act of determining once a year whether your trucks were good to go and ready to deliver the requisite volume of AFFF. These are examples. There is not a one-size-fits-all. There was quite a bit of variability in practice, but here's an example. Equipment testing, this is about trucks. It's not about fire. It's just, can my turret, my nozzle deliver? And that would be an exercise where 3,000 gallons of dilute AFFF was was cast out using these nozzle truck assemblies. That's 341 liters of something that has gram per liter of PFAS in there. You can do the math, and what you see is that if you conduct that exercise once a year, you emit, if you had 3M on board, we're just using that as an example, 3 kilos of PFAS in that single exercise. Now let's say you repeated that exercise over 20 years, because a lot of these activities started to shut down in the 90s, and actually equipment testing kept going a little bit longer. But let's just roll the numbers. 20 years of that activity, you've got a spot that may have 100 kilos of PFAS, and that's just testing the truck and the nozzle. The second one was personnel training. Now this is where they actually brought out the fuels or waste solvents, lit them on fire, I guess it was kind of exciting, and come out with your personnel with about 20 liters of AFFF, 2 liters of the concentrate. Maybe you did that 4 times a year. And what you've got over a 20-year period of just those kind of conditions, 2.5 kilos for equipment and for personnel testing. And so you can imagine if you did this in another location, or maybe it was more than 4 times a year, that mass is building up. And the last entry is an example of a real crash. Now every crash is a little bit different. It depends on the volume used. And so here's an example of a B2 that crashed. 50,000 gallons of dilute AFFF was used. That's 5,600 liters of AFFF. That event alone was a 52 kilo event. We used PFAS as an example, but it wasn't necessarily PFAS. If they used a telomere-based, it would be something else. But the point is the numbers get big in a hurry, and it just depends on how much was used. So the good news is a lot of the equipment testing has really on the decline or been stopped altogether. Personnel training with live AFFFs in these sort of concrete pads and fire training areas, that really started to shut down in the late 80s, early 90s. It still continues in a few isolated sites, but we absolutely store, and the US military as well as the private sector are still ready to use AFFF in response to emergencies today. So just a little more closer look at what was that 3M material. Well, there's the light water famous container in the top, and what we know, it's no longer on the QPL, so we are talking about a legacy formulation. It was made, the chemicals inside this container were made by electrofluorination chemistry. This was a complicated feedstock that went through an organic synthesis process, and what you get is a homologous series. So we like to think about PFAS and Pfoa, but they're only one family member in a whole class with a bunch of family members, okay? And so it's a homologous series, so they're always mixtures. The other defining characteristic of the chemicals made by 3M is they're branched and linear. And laboratories today are very well aware that they now have to be aware and integrating. There's those little blue humpograms down there. This is the signal that analytical labs see. The branched isomers come out in a peak before, not always in that ratio, followed by the linear. And we know that's just the way the 3M chemistry is. And so we have to integrate both of those to create quality data for all of these products. And it's actually a crude synthesis, and we're learning more today about some of the other major and minor components, because we really want to understand the complexity of these mixtures that were emitted in the environment at pretty high levels. So a quick rundown on what is the composition. About 90% are the sulfonates, and of those, PFAS is the majority, the vast majority of what makes up those sulfonates in 3M. Notice the carboxylates are less than 2%. They're probably there as a reaction side product, kind of messy chemistry in, messy out. And about 9.4% are other anions, cations, and zwitterions. And we tend to think of those as other precursors, things that have the potential to biodegrade. And we're interested in what those, and that's where the messy part comes from. That's actually a real complex mixture of materials down in that corner. There is a caveat to this information though. We've only been able to test formulations dating back to the late 80s. We really do not have hard data that helps us understand what was used in the early 80s, the 70s, and dating back to the 60s. And that's what makes understanding what we see at field sites a bit challenging. So what's the other materials? These are examples of the kind of chemicals that exist in those containers today that are good to go and ready in an emergency response scenario. You don't have to be an organic chemist to notice that they're a little bit more complex. You see the highly fluorinated tail on the left-hand side, indicated by the carbon with the fluorine bonds. But then as your eye starts to work right across those molecules, you pick up 2 carbons without any fluorines. That's the calling card of the telomere-based materials. Then you run into a sulfur atom with a red circle around it. I'll come back to that in a moment. And then as you just keep scanning right, you see some pretty interesting molecules. There's some charged states, there's anions, there's cations, there's some oxygens. And these are polyfluorinated forms. Several of these now have aerobic biodegradation pathways. These can be oxidized. However, none of them at this time are on the UCMR3 list. None of them yet are captured by method 537. And so these are essentially invisible to us, but it doesn't mean they're not there. They're very much there in these telomere AFFs. The red S is designating something that can act as a precursor to a family of chemicals we do see a lot of in groundwater. See, we don't see very high concentrations of these in groundwater, and that's consistent with the factors, biodegradation pathways for some of these. What we do see is a telomere sulfonate, where that sulfur has become highly oxidized and becomes a sulfonate, and a very stable molecule under anoxic groundwaters and readily transported. So, what do we do about those things we can't yet see today on method 537, even though we have very capable analytical labs out there working to support the US military and private sectors? There are a few tools that are emerging. The total oxidizable precursor assay is a way to get a handle on the other precursors that are out there. This is now in the literature, and it relies on oxidation, and it takes a sample, and one measures the carboxylates before and after oxidation, and you see the net increase in carboxylates, and you say, aha, there's something in that sample oxidizing. I have precursors, and it's a way to begin to understand what else is out there. Total fluorine by PIGGY, particle-induced gamma ray emission spectroscopy, PIGGY, certainly shorter. This is a new technique where if you can load groundwater onto a sorbent media and then turn that into a solid target and bombard it with protons for a very short period of time, 3 minutes, all of a sudden you get those 2 red fluorine signals, and it turns out it's proportional to the anionic PFASs in groundwater. So that is another tool that's out there, and it's becoming commercialized at this time for trying to understand how much mass is at some of these sites. The third question: what are the sources of PFASs when we want to take a slight step back and put AFFF into a broader perspective? So there's AFFF at the center of this, but remember, it's only 3% of total fluorochem production. If we start in the upper left, we now know, of course, these chemicals come from manufacturers. And manufacturers are not only emitting chemicals for use in commerce. If you look across the top right, and they're intended to be used on goods and products in commerce, but they, of course, also have emissions. Gas phase emissions, they have solid emissions, waste emissions, and they have liquid waste emissions. If we think about the top and humans in the upper right, the top part of this graph, goods flowing that are treated with chemicals come into our homes. They come into this room today. The carpets, the chairs you're sitting on, these are coated for long life and high performance to repel oil and water over their lifetime. And so as these products sit in our home environments, we have gas phase emissions that form inhalation exposure pathways, and also on these materials in their lifetimes give off particles, and we ingest fibers and dusts. And this has been identified by others as important exposure pathways for humans in your own home environment, the school environment, and the work environment. And then you see people in the upper right-hand corner. We now know that residues of PFASs in people are circulating between, say, mothers and infants or fetuses using breast milk and cord blood pathways. Again, well established in the literature. If we go back to the manufacturing and think about, well, where do those goods that are coated, like the articles in this room and the articles in your home, where are they going to go at the end of life? Well, in this country, a lot of them go to landfills. And it turns out landfills is the second most concentrated point source that we know of to date. And that's thinking of landfill leachates in particular, but they're also important gas phase emissions. And that makes sense because 97% of the fluorochem production went into goods that typically fall or go to landfills at the end of life. We now know that modern landfills collect their leachate and they transport to wastewater treatment plants. And so wastewater treatment plants are important thresholds or portals through which leachate as well as municipal waste pass. And we now know that wastewater treatment plants are not good at all in terms of actually removing mass of, let's say, PFAS in Pfoa. And so we have emissions from wastewater treatment plants in the form of biosolids and in the form of effluents that then go on and are applied to agricultural soils or emitted to surface waters. And of course, on the far left, we know that manufacturing facilities, again, emitted their own waste, both liquids and solids, that then enter into these surface soil and surface water environments. And then if we think about the connection between agricultural soils and surface waters, these are important and sources that can impact, say, drinking water sources. And we now have evidence that we have drinking water supplies impacted in this country and around the world. due to this connection with soils and with surface waters. And we know that food is an important exposure route for humans. You'll hear more about that in just a moment. And when you have your fish over here, we do have a few number of spots where there are no-eat fish rules because of that connection between emissions, in this case of AFFF, to surface water. And then you can see the connection back to humans. So what we have is a big, broad sphere in which we're contacting these other sources in your own personal life, even if you don't live next to, let's say, an AFFF site. And the last two sources are chromium electroplating, so a smaller scale, but an important source. So for mist suppression, we know that combinations of carboxylates and sulfonates were used, as well as telomere sulfonates were thought to be substitutes. And so you actually see them used as alternative mist suppressants now. And last but not least, but it's not the focus of this particular webinar, they're very important industrial manufacturing sources, as pointed out earlier. We just list a couple here with the poster PFAS that's of issue, whether it's in New Jersey or New York, here in Colorado, and around the country. And those are very important sites, but they have a different suite of chemicals than perhaps a typical AFFF site. And now we're going to turn it over to Chris and question 4.
Great, well thank you, Jennifer. So I'm going to now address the question of if you're looking at a contaminated site or a site potentially contaminated by these PFASs, what media should be sampled? It's going to not come as a surprise that I'll start off talking about groundwater, particularly at these AFFF impacted sites. And what we am showing here in this slide is a compilation of data primarily coming from Dr. Field's lab, where we've looked at various sites that have been impacted by this aqueous film forming foams and looking at the abundance of these various compounds in the groundwater. And what is important to note is that it is not just the perfluorosulfonates and the perfluorocarboxylates that are the most abundant at these sites. In some cases, we find that the fluorotelomere sulfonates, one of these classes of polyfluorinated compounds that Jennifer mentioned before, can actually be at higher concentrations depending on what type of AFFF was used at particular sites. The other important thing is that at least at the types of sites that we've looked at, where you've had these fire training areas and the AFFF has been released, basically every site we've looked at has concentrations in the groundwater well above the health advisory levels issued by the EPA late last year. And so what's shown here are some highest levels to date, although I'm expecting as more and more sites get sampled, their concentrations, the record breakers could go up. But what you see is it's not particularly uncommon to see up to milligram per liter concentrations of things like PFAS, Pfoa, and the 6,2-fluorotelomere sulfonate at some of these sites. So the concentrations are, again, many orders of magnitude above the health advisories for these particular compounds. And again, when Jennifer mentioned the complex nature of the AFFF that was often applied, when we go and look at these sites, it's not just that we sometimes see these polyfluorinated compounds most abundant, we often see many, many classes. And this is some very simple classification saying, we can see in some sites 7 classes present. I think that number is actually going to go up quite a bit as we look at more and more sites where these compounds are present in groundwater. Of course, the water is going to be coming from somewhere, or the contamination is going to be coming from somewhere. And so it's probably not too surprising, given that AFFF was surface applied, that you might see concentrations of these chemicals, relatively high concentrations of these chemicals, in soils and in some aquatic sediments. And so the figure here on the left is from a study that we did at Ellsworth Air Force Base, looking at a fire training area and looking at the concentrations of PFAS in and around that fire training area. And what you can observe is up to 35-ish thousand micrograms per kilogram or parts per billion of PFAS in the soil. So quite elevated concentrations. What we are learning and what there's a lot of ongoing work to understand this is that we think that the source zone soils can actually be very significant in terms of both concentrations and mass, and that could lead to long-term leaching of these compounds to groundwater. As I said, it makes sense if the AFFF was surface applied, that it would eventually go through the soil and get to groundwater. So there's, again, a lot of research trying to understand to what extent these surface and subsurface soils are really holding on to some of the mass of these PFASs. I'll point out another important study that was conducted by Hunter Anderson and some colleagues at the Air Force, where they looked at non-fire training areas. So it's not just the fire training areas that are impacted. As Jennifer pointed out, there's lots of different types of testing of equipment and crash sites where the HBLF was used, particularly on military bases or other types of locations. And what's shown here is just some frequency of detection at these types of sites for PFAS and Pfoa. What was clear is that PFAS was detected slightly more frequently, not particularly surprising given the HBLF that was commonly used, but we do see Pfoa at many of these locations and other carboxylates as well. The concentrations may have been a little bit lower than at the fire training areas, but in some cases they were quite higher. So as an example of the aquatic sediment, the maximum observed concentration in Hunter's study was up to 190,000 micrograms per kilogram, as compared to, for example, the source area at Ellsworth Air Force Base of only 35,000. So the concentrations can be quite significant in the soils and sediments when HBLF has been applied. One question we often get is, what about the background level in soil? And this slide does a nice job of summarizing two fantastic studies that were done looking at this issue of what is a background level of these poly and perfluorinated compounds in soils from this idea of an atmospheric source. And so this study, went and looked at locations where there wasn't an immediate release. It was not a fire. These samples were not collected from fire training areas, but presumably the concentrations of these chemicals in these soils was a result of atmospheric deposition from various sources in the atmosphere. And the carboxylates and sulfonates, the perfluorocarboxylates and perfluorosulfonates, were the most commonly detected, Pfoa and PFAS being the two most common. But the thing to point out here is the concentrations at most of these locations were typically up to 1 to 2 nanograms per gram, or 1 to 2 parts per billion. A few that were a little bit higher, but most of the data was in that 1 to 2 nanogram per gram range. This is orders of magnitude below the levels that we see at sites where the HBLF has been applied. So it's not that there's not a background level in soil, but the levels in background soils are often quite lower than when we see when the HBLF has been applied at a particular site. As alluded to a little bit before about where these HBLF was released, sometimes it was a controlled release in response to a crash or something like that. There is a potential at some sites for these materials to drain to surface waters. And as not surprised by the aquatic sediment data I presented on a few slides ago, there can be these compounds being released to surface waters. And so it's not a bad idea to sample surface waters, but also things in that surface water. And Jason will talk a little bit later about the potential for bioaccumulation of some of these compounds. But fish are particularly good at accumulating things like PFAS. And it's notable to point out that some sites have provided fish consumption advisories for PFAS because of this contamination of surface water and the bioaccumulation of these compounds in fish. So if you're thinking about trying to characterize a site and there's a potential for surface water to be impacted and fish to be in that surface water, it does make sense to sample and understand what the concentrations are in those surface waters and in those fish. So the next question I'm going to address is, what is the fate and transport of these PFASs once they are released to the environment? The most important distinction I'm going to make in answering this question is the difference between the polyfluorinated compounds and the perfluorinated compounds, as Jennifer talked about before. What we know is that the polyfluorinated compounds, at least for some of the formulations that have been studied of HFLF, can be transformed under various biological conditions to various intermediates, but then also eventually to things like the perfluorocarboxylates. And so this is an example on the right of a biotransformation pathway that's been elucidated, looking at some of these precursors that are in HFLF, forming some relatively stable intermediates. For example, the 6,2-fluorotelomere sulfonate is relatively stable under anoxic conditions, but under oxic conditions, potentially if it was released to a surface water, that can further transform to these perfluorocarboxylates. So it gets at this idea of these polyfluorinated compounds being precursors to the perfluorocarboxylates in particular. And what's notable is that we don't really understand the toxicity of many of these intermediates, but there is some evidence to suggest that the intermediates have some toxicity of their own that might be of concern, even though those are not, for the moment, the primary drivers of risk at many of these sites. When you think about the non-fluorotelomere chemistry, basically the 3M ECF-derived chemistry, not as much research has been done on the transformation of those formulations of the polyfluorinated compounds in those formulations to things like PFAS or the perfluorohexane sulfonate. However, if you follow the analogies from the studies that have been done on consumer products, it's very likely that you would observe transformation to things like PFAS and the perfluorohexane sulfonate if those sorts of precursors are present in those formulations. The most important thing is that everything I've been talking about so far has been about the polyfluorinated compounds. Once you get down to these perfluorocarboxylates and perfluorosulfonates, these perfluoroalkyl acids, PFAS, when you group them together, these are not expected to degrade. Essentially, they are treated as dead-end products and are very, very persistent once released to the environment. There is a little bit of data out there to suggest that some fungal enzymes may be capable of transforming, albeit at slow rates, some of the perfluorocarboxylates, but there's a lot more research that needs to be done to follow that up. The presumption for the moment is that once you get to these perfluorosulfonates and perfluorocarboxylates, they really will not transform at any appreciable rates in the environment. When we think about the transport of these compounds, it's interesting that most of the work has been focused on these perfluorocarboxylates and perfluorosulfonates. So the table here is looking at the various chain length perfluorocarboxylates from short to long, and then at the bottom some short to long perfluorosulfonates, and looking at their organic carbon normalized soil water distribution coefficients. And what the take-home message here is that these compounds generally do tend to prefer binding to organic carbon when it's in the soil or sediment, and that there generally is an increase in that binding or that sorption as you increase the chain length. So the PFAS absorbs more strongly and is more retarded, moves more slowly than, say, the perfluorobutane sulfonate. There is a little bit of deviation from that behavior for some of the short-chain perfluorocarboxylates that were not fully explained as of yet. It's possible that they are not completely binding to organic carbon, and there are other sorts of phases that might be important that will affect their retardation. So the retardation factors, the RF values shown in the final column on the right in this table, are just some hypothetical retardation factors for a particular soil given a certain amount of organic carbon and bulk density and so on and so forth. But it gives you a sense of the relative transport of these compounds compared to water and how that varies among the chain lengths. So you might see not much differential transport. In other words, the perfluorohexanoic acid moves basically at the speed of water, whereas something like the perfluorodecanoic acid is almost 60 times slower than the speed of water. So there is a lot of potential for differential transport of these compounds in the subsurface. And note again, I'm primarily focusing on the perfluorocarboxylates and perfluorosulfonates, which exist as anions in the environment. What's important is because they are anions, it's not just the solid phase chemistry that affects their transport. They also can be affected by the solution phase chemistry. So lower the pH, when you lower the pH in the solution, make it more acidic, that will increase the absorption and retardation of these compounds. And similarly, when we increase the amount of polyvalent cations, so the calcium or iron or magnesium, when those increase in a groundwater or in an aqueous environment, that will increase the retardation of these compounds. There's also some evidence to suggest that these compounds will interact with non-aqueous phase liquids. If there are residual fuels or NAPLs of various sorts that are present at the site, there's some indication that they will interact, although that is very much dependent on the individual compound as well as the solution chemistry in the system. And the important thing here is that if any sort of remedial technology has been employed at a site that would change the solution chemistry or the solid phase chemistry, that has the potential to change the retardation of these compounds. So if you employ ISCO or in situ chemical oxidation that dramatically lowers the pH or increases the amount of polyvalent cations, you could expect greater retardation of these compounds in those systems. Likewise, if you were to oxidize a lot of the organic matter on the soil, that would increase the transport of these compounds because of that altered geochemistry. So you really have to understand not just what the natural background composition is in the solution phase and solid phase at the site, but what sorts of remedial technologies might have been employed at a site to understand how these compounds might be transported. What's interesting is that we have not nearly studied as much the polyfluorinated transport, the transport of these polyfluorinated compounds. And what makes them more interesting than the perfluoroalkyl acids is that they are not just anions, there are also zwitterions and cations. And that's going to lead to differential transport in different mechanisms responsible for the retardation of these compounds. The presumption is that the shorter chain length compounds will generally be transported faster and migrate faster than the longer chain length compounds, and that we expect some pH effects as well, particularly not just for the anions, but for the cations. We expect, for example, that the organic cations, and there's some initial data suggests that these organic cations might absorb very strongly to soils and sediments and might undergo cation exchange processes, again leading to potentially very high concentrations of these organic cations precursors to the perfluoric acids in source zones, and that's something that is undergoing a lot of study at the moment. So to summarize, if we're talking about the transport and the fate of these poly and perfluoroalkyl substances in the environment, it's really important to differentiate the polyfluorinated compounds from the perfluorinated compounds. The polyfluorinated compounds have the potential to transform to the perfluorosulfonates and perfluorocarboxylates, primarily focused on the biological processes, but certainly there are abiotic processes that could also lead to that transformation. And their transport of these polyfluorinated compounds is very much going to depend on whether or not you're talking about an anion, a zwitterion, or a cation. And again, a lot of the precursors we understand may be present as organic cations. When you talk about the perfluorinated compounds, particularly the sulfonates and carboxylates, these are compounds that are not readily biotransformed or biodegraded. They're generally not transformed abiotically as well. That presents a remediation challenge. And they tend to have a relatively high mobility, although that mobility is very much dependent on the chain length of the individual compounds you look at. So again, to wrap up the fate and transport, when you're thinking about these compounds, it's really important to note the differences between the polyfluorinated compounds and these perfluorinated compounds in terms of how they behave in the environment. And with that, I'll hand it over to Ruladeep.
All right, we're going to shift gears to the fun stuff, site characterization and remedial tools for PFAS. So what are some of the challenges and opportunities as far as field characterization? Some of the challenges are a lot of the AFFF sites that are out there have very little or no existing infrastructure. Fuels were used at those sites. A lot of those sites are closed, and they're getting reopened as new sites, and there's very few wells that are present that can be used to further characterize AFFF contamination. The extent, the nature and extent of contamination is for the most part unknown. The analytical methods that we have at our disposal, primarily EPA method 537, which is a drinking water method, and it's the LC-MS method that you heard about from Jennifer. It only gives us a partial picture of the PFASs that are present. You heard from Chris and Jennifer that there are over 300 PFASs. EPA 537 gives us a snapshot of the nature of contamination because we get information on 12 to 16 compounds that are of interest. There's a lot of modified methods that some labs are coming up with that are shorter and cheaper, but each commercial lab is using and offering these modified methods, but the reporting methodology is all based on EPA 537. You heard about the top assay that was developed by UC Berkeley, which accounts for all the precursors, and that's getting commercialized and is coming to a commercial lab, hopefully near you sometime soon. And as far as the current analytical methods that are at our disposal right now, it really depends on the lab and the amount of time you need to get the information, but it takes a minimum of 3 days to get a number because it takes about 2 days to prepare a sample and 1 day to run it with EPA 537. So these are some of the challenges. Some of the best practices for sampling based on the state of knowledge. Again, as I mentioned, EPA 537 gives us 12 to 16 compounds. Depending on the lab, it includes PFAS, Pfoa, and 12 other PFAS compounds. The typical method detection limits are fairly low, about 10 parts per trillion. Some of the best practices for sampling based on the excellent work that the Air Force has done include avoiding Teflon tubing, cap seals, food packaging, and new Gore-Tex clothing just to avoid background concentrations because background concentrations can account for fairly significant levels of PFAS in samples. In addition, we recommend using high-density polyethylene bottles and using QA and QC procedures that include using duplicates, strip blanks, field blanks, and taking background samples. And what we're seeing from the work that we're doing out in the field is always a good idea to consider sending split samples to at least 2 labs to ground truth the results. I'm going to talk a little bit about a field characterization example. This is some of the early work that was done on behalf of the Air Force as they were trying to develop their sampling programs at a lot of their installations. So the first 3 projects that were funded by the Air Force, two of them were led by me, and the scope of work was to define the nature and extent of contamination at point-of-use areas like fire training areas, but also to get a yes-no answer on where PFASs may be present at some of these sites, including fire stations, AFFF retention ponds, and above-ground storage tanks. So what we did at a lot of these sites is we sampled existing wells when they were present. At one of the sites, we had some existing wells, we had 5. At another site, we just did temporary wells. And then collecting shallow soil samples, sediment samples, and at 2 of the sites we had the receiving water bodies, so we collected surface water samples and sediment samples. It is always important when a new compound emerges is to not necessarily reinvent the wheel as far as the methodologies available for sampling. So we relied a lot on high-resolution characterization methods that were shown to be effective for other contaminants. We relied on hydraulic profiling tools, and we did vertical aquifer profiling to get it really accurate, or as much as possible to get an accurate picture of where the contamination is. And for those 2 studies, we sent the samples to 1 lab, and we collected M&A and other parameters to try and see if there's an impact from co-contaminants on the nature and extent of PFAS impacts. This is work or results from Cape Canaveral in Florida. Just a few things that I want to say about this figure. One, the nature of extent of contamination, we were able to get to it after 3 mobilizations. So because there were very few existing monitoring wells, we had to put in temporary wells and to kind of poke the holes in the ground, see what the results, wait for the results to come back, and then either widen our area of interest or shrink it, which is why it required 3 mobilizations. And anybody that does field work knows that whenever you do that, it adds a lot to the cost of a field effort. The star that you see, the purple star, is the former burn pit area at the fire training station, so that's our source area. Right near the source, we saw PFAS at 330 micrograms per liter, and then if you kind of go down within the 100 microgram per liter contour there, you'll see that 720 feet down gradient right before those red lines, which is an air sparge system, we saw PFAS at still fairly high concentrations. Pfoa concentrations were order of magnitudes lower, but again, they were still fairly high. Around 7 micrograms per liter for Pfoa, and they decreased to 4.2, 720 feet down gradient. The plume did migrate towards that receiving water body, which is the Banana River. We collected samples from the river and sent them to Jennifer, and we analyzed them. We found PFAS and Pfoa and the other analytes within EPA 537. We found those in the sediments and in the surface water. And the plume, so it just doesn't stop there. It does discharge. It appears to discharge in the Banana River. But the good news is it is a fairly discrete plume, and high-resolution site characterization methods that work for TCE and other contaminants work well for PFAS, and we recommend that you rely on those types of approaches when you're doing field characterization. In terms of some of the opportunities on the analytical front, in the analytical arena based on this study, what caused us or forced us to go demob and mob so many times is the absence of real-time information. You need to prep the sample for 2 days, run it for 1 day, so you can't really be sitting in the field waiting for the results to come back to chase your plume and to identify your hotspots. So an analytical opportunity is to try and come up with real-time field methods. A lot of organizations are working on developing techniques that are fast, that give you information that is not necessarily reportable, but you can use this information as a screening tool to inform your decision-making in the field. So Eurofins is working on developing a specific compound-specific method that gives you PFAS and Pfoa results at 0.5 ppb levels. There are non-specific techniques like NMR, which can be used for PFAS mixtures. There's the PIGGY methodology that Jennifer has alluded to that is very cheap and gives you a count of or quantifies your fluorine concentrations, and then of course sensors. And the top assay, which gives you a very good picture of that dark matter, which is your precursors. So this is some results from the Eurofins method, and you're looking at a comparison between the field method, which is a direct injection method, so you can run up to 4 samples per hour, and then the fixed lab or the LC tandem mass spec method, and you can see very similar results, which indicate that such tools, when we are ready to go to the field with them, are very promising and are definitely opportunities for us as an industry to invest in. In terms of treatment, what makes treatment challenging is the stability of that PFAS molecule. This carbon-fluorine bond is one of the strongest bonds in nature. PFAS specifically is a very strong compound that is not very susceptible to both chemical and biological attack. So that's one challenge. The other challenge for treatment is the complexity of the PFAS mixtures that both Jennifer and Chris alluded to. So in terms of what's available at our disposal as far as treatment methods for soils, we typically excavate soils and treat them if needed, followed by offsite disposal. There are a number of products out there to stabilize the soils and reduce leaching of the PFAS compounds. There's a couple of products out of Australia that are being demonstrated all over the world. And then other technologies include incineration and soil washing. For groundwater, because of the the high solubility of these compounds, but their low Henry's constant, they tend to like being in the water phase, and air sparging and soil vapor extraction, which are very good treatment methodologies for fuels, they're not very effective for at AFFF impacted sites. Really, there's no silver bullet at this point. The default technology that is sure to stop the migration of plumes is pump and treat. And the default treat part of it is really GAC, although, as if, you know, there's a lot of studies going that are involving different media other than GAC. One caveat with GAC is that we're designing these systems to remove the C8 compounds of interest. So PFAS and Pfoa are the target compounds. So we're not designing these systems to remove other PFASs that are migrating in groundwater that are shorter chain compound. Membrane treatment is also very promising, but it is more expensive. In terms of in-situ treatment technology, SERDP and ESTCP have done a wonderful job in terms of investing into investigations and demonstrations of promising technologies, including PFAS sequestration with alternative solvents, and then proof of concept studies for microbial treatment. So far, over 300 microorganisms have been screened for their degradative capabilities for PFAS, and none of these microbes that are effective for 1,4-dioxane, MTBE, and other emerging contaminants or chlorinated solvents, none of these microorganisms had any luck breaking down PFAS and Pfoa. There is some limited work out of UCLA with fungal cultures where Pfoa degradation was shown using these white rot fungi, but very limited success with PFAS because of that stability of the molecule. The same thing applies with in situ chemical reduction. There's a lot of great SERDP work involving zero-valent metals and bimetals with clay inner layers and co-solvent-assisted vitamin B12 defluorination. This work is being done by Dr. Linda Lee at Purdue University. Some success with Pfoa, more limited success with PFAS. And then there's other experimental work that is very promising, but has not been demonstrated at the field scale, like sonochemical degradation and non-thermal plasma treatment. In terms of advanced oxidation, and lots of promising technologies. We've been hearing about electrochemical degradation. Again, the concern with any oxidative technology is forcing the precursors to convert to compounds of concern like PFAS and Pfoa, and you've been hearing about the precursor issue from my colleagues here. I just wanted to show a very quick case study of precursor formation in the field. This is at Ellsworth Air Force Base in South Dakota, and Ellsworth was the third base where that was funded as part of that Air Force effort to come up with a programmatic approach to the rest of their sites. So it was Barksdale, Ellsworth, and Cape Canaveral, and this work was done by Charles Schaefer. At Ellsworth, PFAS levels and distribution of different PFASs indicated that the historical remedial activities at that site caused impacts as far as the PFAS composition and distribution. So just a bit of history. PFAS was introduced through AFFF use at that site decades ago. Remediation started in 1990 to target co-contaminants like VOCs, SVOCs, dioxins, pesticides, and chlorinated solvents. Remediation happened for over 2 decades. Remedial activities demonstrated at that site include soil vapor extraction, pump and treat, and oxygen infusion wells. So the oxygen played a big role into slowly biotransforming the precursors and causing the PFAS-Pfoa ratios to change. So if you would like more details on this study, there's an excellent paper in ESNT that I'd like to direct you to that demonstrates this phenomena, the precursor formation phenomena in the field. And at that site, precursors accounted for half of the PFASs present. So more research on this precursor issue has been funded by SERDP and ESTCP. This is a study. If you would like details, just look for Jennifer's SERDP project together with David Sedlak at UC Berkeley to learn more about the top assay. And what I want to leave you with here, again, is just a word of caution that our understanding of in situ chemical oxidation right now is very preliminary. The work that's been done at the laboratory scale is promising for Pfoa, but is not really very effective for PFAS. So any sort of design approach involving a field implementation has to account for the precursor issue. So research needs in this arena include a better understanding of the effect of aquifer solids and solutes, understanding the pH effect, and then going to the pilot scale before full demonstration scale type activities. So with that, before we wrap up, we're gonna turn it over to Jason, who's gonna talk about human exposure pathways, health effects, and current regulatory issues.
All right, thanks, Rula, appreciate it. I'm going to talk a little bit about the environmental toxicology and risk assessment and risk management of these compounds a little bit, and then I'll also talk about some of the developing regulatory and policy issues. As Chris mentioned earlier, bioaccumulation is a very important topic for these compounds. And because our analytical chemists are getting really good at detecting these chemicals in many environmental media, and because there's so many different products and potential exposures to humans and other animals and life, it's not difficult to detect PFASs in biological tissues. Many of the PFASs bioaccumulate. As you can see there, I plotted a little small figure from some work we did back in 2008, looking at just the bioaccumulation of these compounds in fish and aquatic animals. And what we found is that the longer the acid, and we're looking at PFOS, PFOA, and those similar acids, the longer the acid, the more bioaccumulative it became. And the more sulfonated, or the sulfonated forms like PFOS were also more bioaccumulative. And what's interesting about these compounds from environmental toxicology and fate perspective is they tend to partition to proteins in biological systems. I deal a lot with hydrophobic compounds like PCBs and PAHs, TPH, that sort of thing. Those tend to end up in fats. We have developed a long history of understanding how those compounds behave in systems and predicting their toxicity and behavior. And now we're kind of completely thrown on our head here, looking at these new compounds that tend to be proteinophilic and end up in protein-rich tissues like blood, liver, and kidney. And another important thing, not surprisingly, as Chris and other folks have mentioned so far, is the acids just don't transform very well in the environment. The same thing with biological tissues. Once they reach PFCAs and PFSAs like PFOS and PFOA, they tend not to transform in biological tissues. Or if they do metabolize, some of the precursors may transform to some of the stable acids. So bioaccumulation is important from a risk assessment and risk management standpoint because if you have animals at the base of the food chain, like benthic organisms, such as like invertebrates like worms and things like that in aquatic systems, they can accumulate levels of acids that are fairly, again, persistent. They're in turn consumed by fish and other aquatic life and transfer up the food web higher and higher concentrations to where they reach receptors like fishermen or fish-eating birds that get a fairly high exposure due to this biomagnification through the food web. And there are several studies that have looked at populations of fishermen, looking at higher body burdens of those who eat a lot of seafood, and really looking at these different exposure pathways. And really this bioaccumulation pathway is really important for a lot of the compounds. In terms of other exposure pathways, looking at human exposure pathways at most contaminated sites that we're dealing with, the major pathways are direct ingestion. Again, I mentioned bioaccumulation, looking at PFAS in the diet, particularly fish and seafood. I didn't bring this up earlier, but plants behave a little bit differently when they accumulate these compounds. Plants tend to accumulate the shorter chain compounds more. So you can also end up with fairly high concentrations in plants. So if they're homegrown produce, for example, if plants are being watered with irrigated water growing in impacted soils, they can also accumulate some of these compounds, representing a potential exposure route for folks that eat that produce. We all know about drinking water. That's been in the press a lot lately. That is also a very important exposure pathway. And then incidental soil sediment and dust ingestion can also be an issue. Most of the compounds we usually deal with at contaminated sites, by the time we reach them, most of the volatile forms in AFFF are no longer present, or they've biotransformed or transformed in the environment. Usually the volatile compounds aren't of main focus. So far in the risk management of these compounds at contaminated sites, we're dealing with the stable acids, and those just don't tend to be very volatile. So inhalation risks aren't an issue. We're not going to be doing a lot of vapor intrusion assessments for these compounds. And then also dermal absorption is usually not an issue, fortunately for the guy in the picture there who's been riding his bicycle through some of the AFFF foam. Drinking water has been in the press quite often in the past year. This Washington Post article from August 9th of last year. EPA is looking at this as part of the unregulated contaminants monitoring program in drinking water sources. And there are, a lot of detections in drinking water sources. Over 6 million Americans have these compounds at detectable levels in the water. We're trying to understand what this means in terms of risk and management and possible effects. And there's a lot of drinking water systems that have been shut down or amended or are under investigation right now because of fairly high levels. Ecological exposures, I'll talk a little bit about that. Many of these contaminated sites have the potential to impact natural habitats where you have fish and wildlife, rivers, streams, creeks, things like that. Again, biomagnification is a big issue, looking at bioaccumulation up the food web. Aquatic food webs are particularly susceptible. You have these compounds that follow the water and may end up in surface water and aquatic sediments. It may be important for fish-eating birds and fish that are exposed to these compounds. Again, in a terrestrial exposure scenario, you may have plants being a very important pathway, route of exposure for plant-eating mammals and other wildlife. Incidental soil and sediment ingestion is also an issue as it is for humans. And again, usually pretty insignificant dermal absorption, unless we're talking about fish or earthworms or something like that. And inhalation is usually not a main exposure route for fish and wildlife as well. So getting some of the effects. EPA recently released several documents last year looking at the effects. And what's known best about these compounds has been derived from studying PFOS and PFOA. EPA has released cancer information and non-cancer reference doses for PFOA, and the PFOS has also been studied as well. We have numbers where we can understand the risk, especially from a drinking water exposure standpoint. And these reference doses are again, fairly low for non-cancer hazards only. There is some information that these are immunotoxic. They have generally developmental effects. So looking at children and developing fetuses, the pregnant mother, things like that, those are going to be key receptors to think about due to the effects that EPA is focused on in their review of these two compounds and the values that have been set. There's some potential carcinogenicity. This is suggestive for both PFOS and PFOA, and possibly for PFOA by the IARC. And there is a cancer slope factor, again, that's been developed by EPA for PFOA. But interestingly enough, the non-cancer developmental effect is actually driving our risk-based criteria. The effects for some of the cancer occur at a little higher exposure than what we see for these developmental effects. There's lots of epidemiological studies out there on PFOA and PFOS, and those have been used qualitatively in support of these values that EPA has developed. In terms of the other perfluorinated compounds that we're finding at sites, there's not a lot of information. There are a few toxicological studies and a few reference doses that have been put out by state and other agencies, but there's limited information. So that's one of the big challenges in the next few years is to understand which compounds we need those toxicity values for, and how do we go about developing them, kind of following the lead of PFOA and PFOS. So I'll put up a few example risk-based screening levels for PFOS in water. And these have been derived from a variety of different sources, the technical literature, the state and federal literature. I'll preface this by saying this is not risk-based. This is not a remediation target or anything like that. These are just for screening to understand potential effects. And what I've done here is separated 4 different scenarios from the left to the right, looking at what concentrations in water would trigger potential concerns for residential drinking water exposures on the left. The second would be concentrations in surface water bodies that might trigger concerns if you have fish consumption, if you have fishermen eating fish from surface water bodies. The third column is some screening levels for direct toxicity to fish and aquatic life. And then the one on the right is a scenario for a fish-eating bird that might be eating fish from a contaminated or impacted surface water body. And you'll notice on the left, I put the y-axis there. That's on a log scale. So we're talking several orders of magnitude here in terms of concentrations where these risk-based screening levels are spanning. And drinking water is not the lowest one. If you look at fishermen risks, those might drive really a lot of concerns in a lot of surface water bodies. So really the take home from this is that drinking water is not the only exposure route we have to think about in terms of evaluating and managing the risks at contaminated sites when you have surface waters that might be impacted with PFOS. And I'll note that, again, most of our analytical detection limits provided by commercial analytical laboratories are generally in the 1 to 10 nanograms per liter range. So some of these levels are actually below that. It's going to be challenging to kind of, again, evaluate and investigate some of the occurrences at sites. And again, ambient background issues are a huge challenge, especially in impacted suburban and urban areas where you have multiple sources from landfills, AFFF application areas, airports, firefighting training areas, things like that. When you have large plumes in groundwater or rivers that are impacted by various sources, it's going to be very hard to tease out and manage risk on a site-specific basis when you can consistently detect PFOS and some of the other compounds as a result of just ambient and background conditions. And again, I'll note that the new health advisory from EPA, that value 70 nanograms per liter, is really just above the upper thresholds of what we might consider ambient or background levels in many of these areas. So we really have very little room for error. And I'll also note that EPA suggests we need to add the exposures, the values for PFOA and PFOS, and evaluate it against the 70 nanogram per liter level. So in terms of risk management, some of the sites with high probabilities of exceeding some of these criteria, some of the draft screening criteria, the health advisories in water and soil and surface water, airports, firefighting training areas, again, places where AFFF is being used or has been discharged over the years, but also our downstream, our waste systems like landfills, sewage storage, land applied areas, wastewater treatment plants, things like that are also important to consider. Petrochemical and chemical plants where these were produced or used, if they had AAA systems, that's also something to take a look at. Chrome plating facilities, it was used quite extensively there. And then again, textile and carpet manufacturers using these compounds as surface treatments, also very important to take a look at those facilities. Some of the risk assessment challenges. It's not just PFOA and PFOS. And you've seen from Jennifer and Chris, there's a wide variety of different challenging chemistries involved here, lots of different compounds, different precursors. As a toxicologist, I feel somewhat confident in looking at PFOS. For the other acids that we can detect with method 537, there's about 8 or 10 of those and a few other precursors. Those are going to be challenging to kind of get a good risk management strategy in place for. And again, there's another 10 to 15 more that some of the analytical chemistry labs can provide information on. And then really high resolution techniques that Jennifer and Chris can do, they can find dozens to hundreds of other compounds at these sites. So really understanding if those present risk and how do we understand and investigate those and give a pretty good evaluation of risk management at those sites are going to be challenging. In part with that, what do we do when we have multiple compounds? We add them together. How do we understand the mixture toxicity of those compounds? I'm used to dealing with PCBs or petroleum compounds where we add them up or we apply certain factors and summing them together and looking at the whole. Are we going to be able to do that with these compounds? It's going to be a big challenge for us. There are also some potential differences in the toxicity and fate of the branched and linear isomers. Again, is that a big deal or not? Is that something we're going to have to dig into? That's, again, an uncertainty right now. Managing risks in terms of background and ambient conditions. That gets into relative source contribution. Right now with the EPA's health advisories, it assumes that a receptor can only receive, a person can only receive a maximum of 20% of the safe dose because we're exposed to these compounds through other consumer products and other sources like fish and seafood and contact paper and coatings and things like that in our daily lives. So really kind of managing risks in terms of background is going to be key. So now I'll switch gears a little bit and talk briefly about some of the ongoing regulatory and policy issues with these compounds. And really what's new on the landscape, recently Governor Cuomo in New York declared PFOA as a hazardous substance. That triggered some emergency regulations and actions to deal with that at some of the sites up there. PFOA will likely be added to the Stockholm Convention, PFOS is already on there. That's basically regulating the manufacture and use of those compounds pretty tightly. Again, the EPA health advisories released in spring last year are no longer provisionary. They are in force. Some of the history of the PFAS regulation, PFOA regulation over the last few years, it really started to emerge in 2001 when 3M discontinued PFOS over the concerns. Very quickly after that, there were some phase outs. There were provisional health advisories issued by EPA in the late 2000s, looking at PFOS and PFOA again. PFOA is basically following along in PFOS's footsteps in terms of being classified on the Stockholm Convention list. And again, it kind of culminating in 2016 with EPA issuing the drinking water health advisories for PFOS and PFOA. EPA has referenced toxicity values, as I've mentioned. The ones that were released in May 2016 were about 4 times more conservative for PFAS, PFOS, and about 10 times more conservative for PFOA than had been previously released. And again, it advanced the idea that, at least for drinking water exposures, we need to sum these exposures. It still left a bit of a question in terms of what do we do about other pathways, and I think we're working through that as a risk assessment community now. Many of the other states have taken some pretty advanced directives on PFOA and PFOS, looking at dealing with those, and there's some variation. Minnesota, New Jersey, Texas have various regulations, so we'll vary a little bit from EPA's approach. For example, the TCEQ, Texas Commission on Environmental Quality, recently released some information on some of the other PFASs. Again, I direct you to that document to see how some of the states are handling it. In terms of Cercla Superfund issues, it's not yet on the list of Cercla hazardous substances. So there isn't a cost recovery mechanism for Superfund just yet. Although it is considered a Cercla pollutant, the contaminant can be investigated at Cercla sites. Again, it's basically the regulatory force behind this is PFAS risk assessments that are being triggered under the Cercla program as part of ARRs per OSWR directives. Some of the other drivers of investigation, public pressure, for example, litigation, Clean Water Act. Some of the work that I've been doing is based on TMDL, or Toxicity Maximum Daily Load, Clean Water Act issues. And there are various approaches at the state level. So just because it's not yet a Cercla hazardous substance doesn't mean that we aren't often looking at it and investigating it actively at many of these sites. And it is enforceable under many different regulatory and legal mechanisms. Again, I'll disqualify this by saying that I am not an attorney. I'm a scientist, so really consult legal counsel because the regulatory and policy landscape is changing pretty rapidly with these compounds. And so now I'll turn it back over to Rula for some closing thoughts. Thanks.
All right, we're going to wrap up with some closing thoughts. We're going to summarize what we told you. So some of the takeaway messages are as follows. Hundreds of fire crash testing mixed waste sites exist due to AFFF impacts. This is just the beginning. There is big potential to reopen sites or create new sites because of the emergence of PFASs. Groundwater contaminated by PFAS used as a drinking water source, as you heard from Jason, is a potential exposure pathway for both humans and wildlife. And we're starting to become aware of the potential of the shorter chain PFAS compounds and their mobility in the subsurface and the difficulty of removing them from water using traditional treatment methods like GAC. AFFF is definitely more than PFAS and Pfoa. We hear about PFAS and Pfoa, especially in light of the new health advisory levels from EPA. They are very important, but they're not the only major PFASs at AFFF contaminated sites. As we discussed, for AFFF impacted sites, a precursor issue is important, especially if we're talking about taking oxidative treatments into the field for demonstrations. As far as characterization, issues and opportunities and challenges, Fingerprinting for sources, AFFF versus landfill leachate versus wastewater effluents is becoming or it's coming on the national scene. As far as forensics, there are existing guidelines and recommendations for sample collection and laboratory analyses, and we'd encourage you to refer back to the FAQ that is going to be a major product from this ESTCP funded project. Real-time analytical tools have a lot of potential and will increase efficiencies while decreasing costs for site characterization, and I cannot stress enough the importance of developing solid conceptual site models as we start doing more RIs. As far as treatment, PFAS is a very stable compound that can make treatment challenging and costly. There is no evidence of in-situ treatment technologies that can holistically address the PFAS issue as of yet. Blindly applying amendments is not wise because of the precursor issues, so we advise caution. There are remaining data gaps on transport and biodegradation that SERDP and ESTCP are trying to address. And then the final frontier is reliance on treatment trains as we start doing more RIs. As far as potential human and wildlife exposure, as you heard from Jason, there are a lot of remaining gaps on toxicity, data gaps that need to be addressed. Drinking water exposure is critical, and paying attention to what the UCMR program is going to come up with as far as guidance is also important. So just to wrap up, there are many PFASs in AFFF and groundwater, sediment, soil, and the toxicity and analytical standards don't yet exist for a lot of these. Treating drinking water sources requires knowledge of target contaminants to identify the appropriate mitigation technologies, and then increasing regulatory and public awareness regarding the presence of precursor and other PFASs is further driving this issue into the spotlight. So stakeholder consensus on the issues and the opportunities and challenges is key. So we'd like to refer you to the project website for additional information. There's a SERDP ESTCP webpage with the project number 2015-74T2, where we will upload a lot of our project deliverables. So with that, I'd like to wrap up. Thank my colleagues and thank you all for your attention. Thank you.